I did my PhD in Alejandra Ventura's group in IFIBINE, that is an institute in Buenos Aires. It depends on the University of Buenos Aires. And it's this building in the Rio de la Plata. It's really nice. And now I'm doing my postdoc in this group with Ariel Weisman, this serious guy you see here, in Fleni. That is this rehabilitation clinic where we have the stem cell lab. And all of these institutes are founded by CONICET also, our national scientific bureau. And this work is about defining network topologies that achieve molecular memory. So first of all, I will define what is molecular memory for us, so we are in the same page. So the idea comes from this set of experiments I will tell you. The motivation is done in cells, in yeast cells. So the normal cell cycle of a yeast cell is it grows until it has a daughter cell, more tiny. But if you have both of these cells, two of these cells sufficiently close, what happens is they stop the cell cycle and start growing into each other, so they have the genetic interchange. So the idea is you can get this pheromone that is the signaling between each other and put it in different concentrations. As what you can see is that there appears a threshold that beyond this threshold, you can stop the cell cycle and make that they grow this, what is called this SHMU. So this other experiment, once you know that, you can do this experiment that is get two populations, different populations of yeast cells, and you give pheromone to only one of each other, but not sufficient enough to go below that threshold. So you have two daughter cells, and then you take these daughter cells, and to one of them, you give it pheromone, again, below the threshold, and the other one give the same amount of pheromone. And what happens is that these daughter cells whose mother have received the original pheromone does the SHMU and the other one doesn't. So this experiment shows that daughter cells can remember that his mother cells received this threshold. So the idea is this is what we call a signaling pathway, that is a network of proteins and chemical reactions. That is the one that senses the pheromone. So these are all in inside the cell, and these one are the pheromone. So in any way, this network is holding the memory of the pheromone. And the idea of this work is to say, how does a network of chemical interactions save information of this stimulus? And how can we quantify it? And how do we compare different network memories between different network topologies? So the idea is to make this quantification. So this is the main talk. First of all, we are defining the quantifier, then apply it to simulated data. And finally, I show some oscillations that have memory. And finally, if I have time, the quantification in real data. So let's start with the quantifier. Let's take this really simple network you can get. That is just one node that has these three different terms. It has dynamics on it. And you can activate it by an input, inactivate it, and it also has a positive feedback. It activates itself. So the idea is if you do an input scanning to its network, it goes by here and comes back by here, so it has hysteresis. And the idea of this really simple network is a good example of a system that has memory, because it has stability, it has hysteresis. The state depends on the history, on if we give a stimulus already or not, or on what was the initial condition, right? Because if we start here, it ends up here, but if we start here, it ends up here. So the idea of all of these things is what we want to hold in our quantifier. It's the same as when you have a memory device. It's a physical system that stores memory, in this case, in the hysteresis of the magnetic cells. So the quantifiers go like this. Imagine you make an initial condition scanning. So you have all these different curves, and each one of them to an initial condition. And you measure this quantity. This quantity is the slope of these curves at a given time point. And then you make another derivative in terms of the initial condition. You go this way and see the changes of slope. So This tells you if these curves are diverging, are parallel, or are convergent according to the sign. You can make a double integrative, and this quantifies how much separated the two dynamics remains in the time. The two dynamics are the two different curves according to the limits of the integral here. Yeah, and how much you can distinguish between them if you measure a final condition and you have waited all this time that is here in the limits of this integral. So the idea of the quantifier is just the double integral of this, that gives you just the difference in terms of the initial condition. But as we want to know how a network can measure memory of some stimulus, the idea is that the stimulus has to end because we want to measure the memory. So you just take the initial condition as the condition, as the state of the system when the stimulus ended. So you can pass from this to this, where A0 and A02 are the two different stimulus. So this first stimulus, I1, will be our control stimulus, and in this case, just not giving a stimulus to the network. So for example, in this network, we apply the memory quantifier. And as you can see, you give some transient stimulus. So this network gets activated, and because of the positive feedback, remains activated, and you can measure just measuring the difference between these two cases. So now we apply our quantifier in order to know what kind of network motifs, what are we calling, what kind of different combinations of networks are capable of holding this memory. So the idea is we take all the combinations of activations, inactivations between these three nodes and between themselves. And the idea is to scan all of these combinations with 10,000 parameter sets for each one to make a complete study, and give the stimulus, both stimulus to each one, and search for this memory motif, this memory mechanism that appears here. So what is a motif? A motif is like, you can think it as a minimal computable processor. It's the minimal thing, a combination, a set of 3 or less or more nodes, but little ones that have the capability, that provide a higher network, a capability of process information in some way. For example, give an updation. If you get this motif, you have a direct activation here and indirect inactivation here, and this makes this pulse that if you give a stimulus to this green node that is sustained, the response is not sustained, it has a peak. So this system can adapt to something and sense, for example, changes in the total stimulus and not the absolute value. So here are the integrated networks, 1 node, 2 nodes, and 3 nodes, all of them. And we make the resolution for each one. And we have this data set, a final. As you can see, this is the final memory vector we can have, where each number represents a parameter set of this particular combination. And it has the memory of this node, and this node. If it's positive, it implies that that node activates irreversibly with the stimulus. If it's negative, it inactivates irreversibly with the stimulus, which is also memory. Or if it's 0, as in almost cases, it hasn't got memory. So we find, of course, the original network example for 1 node. For 2 nodes, we have these positive feedbacks, 1 node and 2 node. For 1 node, A has a positive feedback and activates B. So it remains both of them activated. Here, in the 2 cases, A activates B and B activates A. So A is activating itself. And again, it has memory. And we also find these 2 ones asking that A activates, but B inhibits with the stimulus irreversibly. So you have these 2 cases, and as you can see, there's an active-inactive node symmetry that when you ask for nodes activating to inactivating with the stimulus, you end up with the same motifs, but only changing signs of all the edges that are in the node you are asking for change. Oh, sorry. I think I don't know what's going on. So for 3-node networks, again, we have a 1-node positive feedback, always with the A, because it's the node that receives the stimulus. And here, in this case, it activates directly or indirectly the other ones. For 2-node positive feedback, you have all these 4 combinations. And you also can have a 3-node positive feedback, clockwise and anti-clockwise. And again, if you ask for these 2 nodes, for example, to be inactivated irreversibly with a stimulus, you have all these combinations that are just the symmetrical, changing the sign of the arrows of these 2 nodes. And also, you have these 2 panels with the intermediate states that give exactly the same motifs. And also, there's another symmetry here of interchanging the violet with the blue. So this says that our method was really robust because we find these motifs are not any more and not any less, and they are all symmetric, so this is good. So this idea of active-inactive node symmetry can also be generalized and applied to another feature, dynamical feature you want. For example, in this case, you take the motif I told you before, the adaptation one, and you can apply this active-inactive node symmetry and compute this curve, and then go to this other motif, just applying this symmetry, and you end up with a dynamic that is similar, but like inverted. So it also activates. Thanks. So I told you now about another kind of motifs that can also have memory. For example, in the oscillations, we can measure with our quantifier and detect that these cases has oscillations too. Imagine this motif oscillates, so there's the oscillations, and then you give the stimulus, and what happens is, because of this positive feedback also, you end up from oscillation to a fixed point value. So also have memory, because if you measure this difference, you are measuring something different from 0. But what we ask here is, okay, you can have memory without 2 different stable points, but can you have memory without a positive feedback? Well, if you take this other motif that it's called a repersilator, you can also oscillate. And for example, you make this computation, and you have these dynamics. You again give the stimulus to both motifs that are equal, are oscillating. And then you end up, again, with both oscillations, is similar at this part. The final part is similar at the beginning part because there is no positive feedback. But what happens is that you can measure a difference in the phase with our quantifier. So our quantifier says that this system also can have memory, but now it turns in the fixed point. The final fixed point has changed before the stimulus, after the stimulus. So here, the physical storage of the memory is the difference in phase. And well, finally, I want to go rapidly through this. That is the application that we did in the lab with using stem cells that are embryonic cells from here. And we studied the transition between embryonic to the mesendoderm cells. That is called INEU prime. But the idea is to give the same stimulus to cells, a transient stimulus of 1 or 2 days of this differentiation. Take an embryonic stem cell and say you have to be some adult cell. And the idea is to apply the quantifier, and we measure that effectively these cells remember the differentiation stimulus, and if you give a differentiation stimuli to the cells and take it out before 24 of 1 day or 2 days, they continue the differentiation, and that doesn't come back. And that's the idea. So the conclusions of this talk, we define a memory quantifier applicable to any network, any topology, any edges you can have. We found minimal motifs that can provide memory to a network, the positive feedbacks. And oscillation systems can also remember a stimulus and give positive to our quantifier. And finally, we tested in mouse embryonic stem cells. So if you have any questions, please feel free to ask.
Okay, we have time for a short question. Yeah, please. Yeah.
Thank you for your talk.
Thanks.
Maybe my question is too basic. I don't know much about it, but very interesting,
Yeah, of course.
everything.
Yeah.
Does this type of influence only works on directed networks, or can you have topologies in undirected networks that also present memory? Because you need flux, right? In your analysis.
Yes, I never think about how can we apply our quantifier to undirected networks because of I am thinking on dynamics, and in dynamics you have one node is activating or inhibit the other one. But yeah, maybe.
In a system where you don't have inhibition, just activation, is it also possible to have memory?
And it depends. I suppose it depends what the edges are and what do you want to quantify. Because if you have a stimulus to the network, you have an arrow there. The stimulus is activating or is doing something to your network in a directed way. And this where quantifier the memory to this stimulus. So maybe you can do something analog. I never think about it, and we have to see in any network how can we do it, but we can talk.
Okay, thank you.
Okay, let's thank Federico again.
Thank you.
Now is Leonardo around?